Hello students, welcome to EPG Pathshala. I am Chaudhary from Jawaharlal Nehru University, and today we are going to talk on the module database searching under the paper Computational Biology. Now what about database searching? Now as you are well aware from the previous lectures, biologists are collecting a huge amount of sequences from different organisms and they are putting them in computerized databases. Now other biologists, other users of this database, they will have to search for a particular sequence or a group of sequences out of that database. And how does one do that? In this lecture, my objective will be to take you through, to walk you through the different techniques and methods that are available to us to search databases. Now basically one can divide the database searching methods into just two groups. Number one, text-based searching, and number two, obviously sequence-based searching. In text-based searching, you just put in any keyword of your choice. If that keyword or a similar keyword exists in the database, it's going to be returned. It's exactly the same way you do a Google search in the internet. There are, of course, for sequence databases, specialized interfaces like OnThreads, for example, but all of them work almost in the same way as Google does. So there is nothing very different out here. The other technique, sequence-based searching, is the one which is specialized for biological sequence analysis. In this mode, you given a sequence and a similarity value of that sequence within all the sequences which are stored in the database, and that could be millions of sequences. All of these are searched, and the ones that are similar to your query sequence, by some means, by a well-defined measure of similarity, only those sequences will be returned. Now, there are very many ways to do this. There's an algorithm called BLAST, an older algorithm called FASTA. There's another one called Smith and Waterman. Now, obviously, given that the databases are very large, we need to do this very fast. So the faster algorithms, one of the newer and the faster algorithms is called BLAST. And here onwards, most of my talk will be centered on the BLAST algorithm, and I'll go ahead on that. Now, when you are searching a database with sequences, the one question comes to mind first of all. Are you going to search with DNA sequences? That's how most of the sequences are stored because they come from gene sequencing methodology. Or are you going to search on the protein sequences? There are, of course, very few, not less than 1% maybe, sequences of proteins are directly sequenced from at the protein level. Majority are comes from the gene level. But then one can always translate a gene sequence into a protein sequence and we can have databases. The question comes now, should we search at the gene level or should we search on the protein level? Now if you want to search at the gene level, now there are the alphabet size, that the alphabets, the DNA alphabets which make a gene, there are only 4 of them, A, T, C and G. And the size of the gene would be about 3 times larger than the protein sequence, right? Now in such cases, if you give a query, just say some sequence A, C, T, G, the chances that it randomly matches somewhere in some gene, someplace in some gene, somewhere in the database, without having any biological significance whatsoever, is rather large. On the other hand, if you look at the protein alphabet, that has a size of 20. We have 20 amino acids. Now if you give a random query, the chances that a random query of length 8 or 10 matching something in a protein sequence database, which has 20 possibilities for each position, is much lower. So therefore, searching databases using a protein sequence is much more specific. There are much less chances of random hit. Okay, given that the protein sequence is, searching on the protein sequence is much more specific, sometimes what we do, that we translate the databases which are at the DNA sequence level to protein sequence level. We can even translate our query, which might be a DNA sequence, into a protein sequence and then search at the protein level. This increases specificity. But then again, there might be a situation, there might be a certain biological problems which requires you to search at the DNA level. So barring those specific situations which may arise, generally searching on the protein sequences is much better because they are much more specific. So going back over the whole thing, that translating a DNA sequence into a protein sequence causes some loss of information, of course, because there might be synonymous codons which just code for a single amino acid, so you lose that information. But protein sequences having a larger alphabet and being more conserved give very specific results. The chances of random hits are less. So in general, in the most common cases, it is better to translate a DNA sequence to a protein before searching, but there might be exceptions. Now the second point which I would like to reiterate at this point is that there must be certain criteria of what makes a search algorithm a good search algorithm, or maybe some other algorithm not so good. So the quality, the performance of a search algorithm, whatever that may be, we can judge at two levels. The first is called sensitivity, which is the ability to detect a true evolutionary relationship. The more sensitive a technique is, it is able to detect more distant evolutionary relationships. That is, sequences which are which have diverged millions of years ago. Even those will be detected by a sensitive technique. Specificity, on the other hand, is the ability to reject false positives. Sometimes when you give a query, it returns you a hit, but that has no biological or evolutionary relationships. It's just a random hit, something false positive, that's what we'll call it. So the idea of specificity is that a technique which rejects false positives. So the more specific is a technique, the more reliable will be the result. Very unfortunately, sensitivity and specificity, they tend to go different ways. So if your technique is very highly sensitive, chances are that it also gives you a lot of positives, if false positives. On the other hand, if your technique is very highly specific, very highly reliable, chances are that you miss out a few things because it is not sensitive enough. So given these ideas, let's come into the technique of BLAST. It's an acronym, not just a word, and it stands for Basic Local Alignment Search Tool. It was invented by Carlin and Archul around 1990, and the great thing about BLAST was that it increased the speed of searching databases compared to an older program called FASTA, and it also gave better quality results. And it integrates a substitution matrix. In the alignment on multiple alignment, in the module on multiple alignment, we did discuss substitution matrices in some detail. And unlike the previous program called FASTA, BLAST uses a substitution matrix in the search process itself, which makes it give you better quality results. And as of today, I can say with quite some confidence that BLAST is the industry standard tool. Almost everybody would come and use BLAST. The other programs which were there before BLAST are hardly ever used. So from now on, the discussion is going to be mostly on BLAST. Now, even though we think about BLAST as a single algorithm, but there are very many implementations of this algorithm. So this results in a number of programs. Let's call them the BLAST family. So for example, we have BLAST-N. Now this particular program uses it as its query a DNA sequence, and the database which it searches against is also a DNA sequence, and the comparison is done at a DNA level. Now I said earlier that sometimes protein sequence level searching is better, but it is not to say that DNA sequence level searching is not ever done. And if you want to do it, then you should use the BLAST-N program. Then there is another program called BLAST-P, which is the protein counterpart of BLAST-N. That is to say, it uses a protein as a query, its database is a protein sequence database, and the comparisons are done at protein level. Then you have the program called BLAST-X, where you use a DNA as a query and your database is protein, which means that even before you start your search, your query DNA sequence is translated into a protein sequence computationally. Now, please note, it doesn't have to be that your query DNA sequence is exactly a coding gene in reality. It doesn't have to be. whatever it is, you can, given a DNA sequence, you can always translate it into a protein sequence. You do that and you search a protein database. Why? Because, as I had mentioned before, that searching a protein query against a protein databases gives you more specific results, less chance of random hit. So therefore, BLAST X over BLAST N gives you more specific results. Then you have T-BLAST N, where a query is DNA, the database query is a protein, database is DNA. In this case, the database is pre-translated to protein sequences. So your query protein can be translated against a protein database which has been translated from a DNA database. So the search is carried out at a protein level. Again, more specific, like BLAST X or BLAST P. And then again T-BLAST X, where both the query and the database, both are at DNA, but both of them are translated to proteins before you start the search, and the search is done at a protein level. So again, you go for specificity. As you can see, out of the 5 programs here, 4 of them search at the protein level. They give you more specific results. 1 of them searches at the DNA level, gives you more random hits. But there are specific situations in bioinformatics where sometimes that has to be used, and that is why that is there. So now, going into the BLAST algorithm, we need to first define ourselves a few terms. Without them, we are kind of lost. So the very first term I come is a segment pair. What is a segment pair? Given 2 strings, I have 2 substrings of the same length which can be aligned to each other without a gap. So if I have a large string, say P, and another large string Q, if there exists a substring S1 within P and a substring S2 within Q which can be aligned without a gap, then that would be called a segment pair. Now then there is a locally maximal segment. Now what is that? It is nothing but a segment pair whose alignment score without gaps, that is, because a segment pair is a gap-free alignment, cannot be increased any further by extending this particular length of the segment either way, on the left side or on the right side, towards the 5' side, N-terminal side, or the 3' or the C-terminal side. So you start with a segment pair and then you extend on both sides and you see if the alignment score increases or not. At some point comes in where it is reached its maximum, cannot be increased anymore, then that particular segment will be called a locally maximal segment. Then of course you have the maximal segment pair, or MSP. That is, given 2 strings S1 and S2, that particular segment pair which has the highest score among all the locally maximal segment pairs that there are there. And then finally you have the high scoring pair, that is when a database is being searched, okay, the BLAST would attempt to find a maximal segment pair, or MSP, for each sequence, and the entire database, the highest scoring MSPs, would be called HSPs, right? And so that means for every sequence I can find out an MSP and I score them. Now in the entire database there are millions of sequences. If I do this to the entire million of sequences, I get million MSPs. Those which have the highest scores above a certain threshold which you can set, those will be called HSPs. The HSPs are the output of the BLAST program. And what you get when you search a database using BLAST are the HSPs, their alignments and their alignment scores is all that you are going to get. So how do you go about it? Now what is the process? So what you do, when you start the algorithm, you take a length parameter W and a threshold score T, and then you find all segments which are W length and which has an alignment score without gaps greater than T. Such kind of word pairs, one from your query and one from the database, these are called hit. Now the recommended word size is 3 to 5 for amino acids and around 12 for nucleotides. Now why is it smaller for amino acids? Because there being 20 amino acids, even with length of 3, that's just a tripeptide, the chances are that you would get a random match with that tripeptide is 1 in 8,000. But for DNA, if we use a tripeptide, the chances are that you would get a random match is just 1 in 64. So you go and increase the length and you go around up to 12, thereby reducing this chance. So if the threshold alignment score T is increased, the number of hits are of course reduced, and your program runs faster, gives you more specific results. However, if we reduce the t, you can get even distant relationships where the homology is very far away, but the chances of random hits are now much more. So now once you have found a hit, what do? You go and extend the hit, that the hit is the segment pair. You extend the hit on both sides until each time you keep on calculating the alignment score. Now one would expect that as you are extending the hit, the alignment score may increase. If it increases, continue extending it. If it does not increase, stop. So what you get at the end is that particular length which gives you the maximal alignment score in that local region, and so we will have the locally maximal segment. Now why is it that the score can increase, can decrease? Because when you are checking that alignment scores, you are using a substitution matrix, and as we have already discussed in case of local alignments and in case of multiple alignments also, the substitution matrix can have both positive and negative values, and therefore the alignment score can sometimes go up, sometimes go down. So that is why all that I'm trying here is that I go on extending my length until I do find that particular length which gives me the maximum alignment score. And the moment it starts reducing, this extension process is terminated, and whatever is the maximum alignment I have gotten, that is my locally maximal pair. What BLAST does is that for every sequence it finds out the LMPs, they have their alignment scores, and those which are very high, they are defined as HSPs, and these are output, right? So now this output is now formatted in a very specific ways and depends upon the users. Different users might want this output in different ways, and the best thing about the BLAST program is that it can give you the output in the way you want, and then you take it. So let us look at the different ways in BLAST gives its output and what do they mean. So the first thing is the traditional report. Okay, this is the most common thing when you first use BLAST. Probably this is the way you will get it because this is the default mode of output, right? Now this traditional report, it consists of 3 sections. At the top, we'll call it the header section. It contains information about the query sequence and the database you are searching. You might search all sequences in the database, or you might search some particular subset of the database according to your need, and all this information, whatever you are doing, this will be given in the header. Then there is a one-line description which provides a very quick overview of your search results, and after that the third section with the detailed alignments which of the HSPs which BLAST has returned to you will be given to you one after the other. So let us look at these 3 sections in detail one at a time. So here is an example of the traditional output. So this is the header portion, as you can see. So you have the particular program, the day you have searched. These are written right at the beginning. A reference to the BLAST program is given, and I would strongly recommend somebody to look up and read that paper. Then you have an ID number, that RID, and this huge number after that, which you can use to retrieve your results even at some later time. So you don't have to sit in front of the web page when you are doing a BLAST search. If you just take out that ID number, and then you can use that to retrieve it even later. Then a little bit of a description of your query protein. In this particular case, it is the RAB protein, as you can see. It has 656 amino acids, and that's written over there. And then the particular database, what we used in this particular example is the non-redundant SwissProt database, and at the point of time when the search was done, it had that many 102,387 sequences with that many bases or amino acids in that sequence, in the total over all the sequences. And then of course, if you have any trouble with any questions with the results of the search, it refers to you a FAQ, or a frequently asked question set. And then additional things, as you can see, there is a link to the taxonomy reports and etc. So you can click on those and you will get those additional information, the help, etc. etc. Now if you go back beyond the header portion, now this part is very interesting. What you can see at the top is a color key for the alignment scores are given. Now the red color is with the highest scores, while the magenta, the green, the blue and the black, the scores gradually come down. And then just below it there is a scale bar. Now our protein, as we saw in the previous slide, was about 656 amino acid, so the scale bar is going slightly beyond 600. And then those lines which are there in various colors, each line represents one sequence in the output. Now the first three lines, the first line for example is red almost throughout. That means that represents a particular output which matches your query sequence throughout the length of the query sequence with an extremely high score. The same thing is about the second and the third. But then if you look at the fourth, we see that the length of the match is now somewhat reduced. It's even less than 600, maybe around 550, and different parts of the score, that output sequence, you'll see that they are colored differently, and one of them is magenta, part of it is green, indicating that these are the segments we are matching at that score, which is not that high. And this goes on, and at the bottom you see those black lines here and there, and which means these are probably random hits. That is, set of sequences, a small part of it now is matching with a query and giving a pretty poor score. So most of the time we'd be concerning ourselves with the top level. Now if you move your mouse over the top sequences and then a blurb comes out which gives you a little bit more description, but in any case the entire description is now given just below this in the third section of our output. This is the final section of the output, where on the left is the key to every sequence. Now that's a link which you can click and that will return you the sequence. If you notice, something called GI and then a number, SP and a number, etc. are given. Now these are the accession numbers of the database. GI stands for gene index, SP stands for Swiss prot, and so on and so forth. So this is how the sequences are identified in the database, and they must be somewhat similar to your query protein. The second part, written in black over there, gives you a one-line name or a little bit of a description. The first one I could see is a RAB protein, it's something geranyl transferase or whatever. So then the third column gives you how similar it is. It's a number. Now this is the score in bit unit, and the larger the number, the more is the similarity between your query and your target sequence in the database. We will have more to discuss about the scores. And then the fourth line is an E value, again something we'll call an expectation value, which is, for the time being, let's look at it as a normalized scale. In this particular case, the smaller the number, the more similar your query is to the target sequences. As you can see, the first three numbers are 0, which means essentially those proteins are absolutely similar to the target. Thereafter, the numbers gradually increase. As the similarity goes down, the E value goes on increasing. We'll have more to look at the bit score as well as the E value in the subsequent part of this talk. And finally, you get, if you click on any one of them, and then you will get the full alignment, the full local alignment. That is the part of your query which is matching the part of the target. Now you have the query sequence at the top, and you have the target sequence or the subject sequence at the bottom. In between is how similar it is. If they are identical, then the amino acid itself is put in. If they are similar, then the plus sign is put. If they are not similar, then nothing is done. So that way it goes on describing the entire alignment for every sequence that is there. And mind you, there's quite a lot of results. There's a lot of calculation out there. But the beauty of the BLAST program is that it can do it over a few seconds at most. Now this is something I think one of the biggest achievements of the 20th century, that in the National Center for Biotechnology Information, NCBI, which is in Bethesda in Maryland, they have these computer servers which is serving you the entire world with this BLAST output for free. Now imagine how efficient is the algorithm which is doing it. I can search, you can search, anybody across the world is searching, and all the searches are being done in the one and the same place. And they are catering to the entire world, and they're catering for free. This would not have happened if the resources which they have to put to do so. Of course, that is a huge amount of resource, but something which they can afford. And that has happened because of the efficiency of this algorithm. So just the fact that the entire world is searching more or less in a single place tells you that the algorithm to do this search must be fantastically efficient. And why is it so? What makes it fantastically efficient is something which will go. In the meantime, let's look at the other kind of outputs. Now, many times, you are not just a simple user of BLAST, you are a more sophisticated bioinformatician, and you would like the output to go into some other program, some further analysis you are going to do. Now, if we saw the traditional report, it's very easy to read, but it's very difficult to parse. So this particular form of the output, so-called hit table output, gives you in a tabular format, which you can read it with Excel or any such spreadsheet or any programs that you are tending to make. And if you want to do further analysis on this, those analysis can be easily done. So this hit table output is more tuned to those kind of users who are running BLAST not just for the sake of BLAST itself, but they are going to take that output and do further downstream analysis. It simply makes this analysis easy. So in summary, the hit table form is a concise description of the BLAST result, not as detailed as the traditional output, and it is easily and automatically parsed. Here again, the lines starting with an hash or comment lines are generally ignored by the parsing programs, and the rest of the lines are separated into structured fields, which are, even a novice programmer can write a simple program to parse and read them. So whenever you want to do automatic analysis, you can do that. Talking about parsability, computer scientists will tell you that if you want parsability, you should use a structured output system, something like XML, extended markup language, or anything. Now BLAST uses its own form of XML called ASN1. It can also use the traditional XMLs. So both of these are there. So sophisticated programs can read these output forms, ASN1 or XML, and these are what, if you want to do downstream analysis, this is what the way to go on. So now, as I just indicated a while ago, that the great thing about BLAST is its extreme efficiency. So the point is, how is it that it has become so efficient? To understand this, we have to look at some statistical properties, not just of BLAST in particular, but of global alignments to start with, then local alignments, and go on. So if we consider a global alignment and there is a particular score, let us consider many two sequence global alignments. I should get and maybe randomly picked up sequences. Some of them have biological similarities, some of them do not have. Let's just consider them all together. If you look at the distribution of that kind of scores, what will it be? Okay. So the expected global alignment score for random sequences is known to increase with the alignment length. The longer the sequence, the more is the score, even if it is a random. However, the rate of increase of the score with the length of the alignment turns out to be very difficult to predict, and that's an unsolved problem as yet. Not very clear exactly what should be the rate. However, it is also known that the alignment scores for random sequences depends upon the alphabet size, the frequency of different characters in the alphabet, and so on and so forth. There are quite a few things which are important. So what one can do, as used to be done by previous programs, because there is no clear mathematics out there, so one could approximate the distribution by doing some empirical analysis, doing some simulations. So what one can do, I just get two sequences, get an alignment scores, I shuffle the sequences many times, thereby randomizing them and seeing that how different is my test alignment score from the alignment scores which I obtained randomly. Now this was the principle used by, for example, the FASTA program to check for the significance of your alignment. But this is where time was taken and it slowed down. BLAST beautifully does away with this process, and in fact it uses a simple analytical formula to find out how significant the score of your alignment is. So one does not have to do any simulation systems. Okay, let us look at the statistics of global alignments. The statistical significance of a global alignment score can be estimated in the form of what we call a z-score, defined as z is equal to s minus mu over sigma, where s is the alignment score of a real pair, while mu and sigma are the mean and the variance of alignments obtained from randomly shuffled sequences. So we have this z value. Now if the alignment score of a real pair is something close to random, which means that it's not very different from the average value of a random sequence, which would be mu, so this z-score would be very small. But if it is a significant alignment, if it has a score which is very different from the average of random alignments, then the z-score would be very high. So let's start with that. Now, while the z-score is very commonly used in many fields, it is not really a valid measure of statistical significance, because it will only work if the distribution of the alignment scores of random sequences follows what is known as the Gaussian or random distribution, and that there is no guarantee that that is indeed true. In fact, it is not true. If you look at for local alignment scores, particularly in the case of ungapped local alignment, then the scores are known to follow a particular kind of distribution. It is not the Gaussian distribution we refer to, but it's called an extreme value distributions. And as was shown by Carlin and Altschul, that in case of a database search, it was proven by them that the score of the expected number of HSPs with an alignment score of at least S is given by that formula, that E is equal to K M N e to the power minus lambda S. Now notice where S is the particular score and while E is a so-called expectation value. K and M and N are constants, and where M and N are the lengths of the two sequences which are being compared for the local alignment, while K and lambda are two parameters which sort of sets a scale for E value and the distribution of the scores, and it depends upon the kind of database, the diversity of this K and M depends upon K and lambda are two parameters which characterize the distribution of the scores, and it can be thought of as natural scales for the search space, and they depend mostly on the alphabet size for DNA alphabets, for protein alphabets, whatsoever. Now these can be found out a priori and these numbers can then be used. So what we get finally from this equation is the E value for a score, the expectation value for the score given S. So now the raw scores again, they don't have much meaning without a detailed knowledge of the scoring system. So to make the scores independent of the scoring system, the raw scores are normalized and we get a bit score or S prime, which is again given by S prime is equal to lambda S minus natural logarithm of K times the natural logarithm of 2. In terms of the bit score, if you look at the E value, then the E value is given by M times N times 2 to the power minus S prime, where again M and N are the lengths of the sequences. Now what exactly that the expectation value means, statisticians will tell you something of course, but for our level that we can say that the probability of finding at least one HSP which is a score greater than S prime is given by the last formula, P is equal to 1 minus e to the power minus E. Now, so this is what we understand. So given the expectation value, we can calculate the probability that I can find out one HSP with a particular score at random. Now if this probability is rather high, which means that the chance that the hit which I have obtained can just be obtained by randomly picking up some sequence from the database is rather high. So that is unlikely to be biologically significant. So what I want is when the probability is very small. So which means that I get a particular hit score and the probability, the chance of finding it simply by picking any sequence in the database with our eyes closed, if that just does not happen, that probability is very small, which means that definitely the alignment which I have gotten must have significant biological importance. So lower the probability value, higher is the biological significance of a particular alignment. And if you look at from that formula, lower the probability will come when the expectation value is also low. So if the value of expectation value is 0.01, then what we can expect roughly, that there will be one true hit out of 100 randomly picked up sequences, right? If it is 0.001, then you have to pick up 1000 times randomly to get one hit. Definitely that should have good biological significance. In practice, we use expectation value, cutoffs of expectation value, which is less than 1 in a million, that is 10 to the power minus 6 or 10 to the power minus 8, where the chances of getting that kind of alignment is like extremely small or negligible. Therefore, it must be biologically significant because it cannot be gotten at random. On the other hand, if the expectation value is about 5, then in every time we search, we should get 5 random hits. So remember, the lower the expectation value, the less chance of a random hit. And now what should be the cutoff? It depends upon what kind of problem you are looking. But in general, typically, the expectation value cutoff is set between 0.1 or 0.001 or something. So if your value is anything greater than that, unlikely that your results are significant. But if your value are less than, let's say, 0.01 or 0.001 or something, then it might be significant. Remember, at the end of it all, it only suggests the chance that your alignment is significant or not. It will never tell you whether it is indeed significant or not. What you are running is a software program. It is not doing your biology for you. So it only tells you what is the possibility that your alignment has a biological meaning. It will not tell you the biological meaning. That is up to you to decide. Now, having said this, is that the only thing that we can do? No, certainly not. There are very many advanced versions, very many extensions to the BLAST algorithms. Some of them go by the name of Mega BLAST, Discontinuous BLAST, Phi BLAST, Psi BLAST, RPS BLAST, etc., out of which some of which I will be covering in subsequent modules. Thank you.